what we had learned of importance in that up to now, or just had learned, is a very different view from how that I've ever seen of how variation is created. Okay. That there is external mutation, for example, cosmic rays, and they create mutations for sure. Okay. And the old view would have said, and then those mutations lead to offspring where external natural selection acts upon them. But that's not actually right. Any organism that is relying on that is likely to be extinct. Rather, what happens is external mutation undergoes internal repair. And it's not, oh, maybe it does, maybe it doesn't. The cells in your body spend huge amounts of their resources, energy and molecules, to accomplish this repair of external mutation. And then they do this surprising thing, at least at first glance surprising. They start to mutate themselves. And it's really those mutations which are going to undergo natural selection. So the creation of offspring is not some random event where mutations occur because of external things. It is, one could say, a recognition by the cell that it both has to adapt to changes in the environment, and that means it has to mutate. But it's going to do it in a very intelligent way. It's going to program its own mutation. And we have this, in general, proposition 3, variation is primarily the result of processes carefully programmed by prene sets. Okay, so the cell is computing something, and it's computing what it should do to mutate itself. All right, so now we're going to move on to HIV. Hang on a second here. You can see this, hopefully?
Yes.
Oh, good. All right. So we're going to see how HIV has learned through evolution. How did I put it here? To create variation through the use of carefully programmed intentional internal mutations. These things. All right, let's get into it. So this is the first pass through this material.
Intentional mutation in HIV. To understand HIV's use of intentional mutation, let's start with a sports analogy. You own a team that has just won the championship. Do you trade some players? The initial response is no, don't touch anything. But is this the wisest thing to do? You can be virtually certain that next season will not be the same as this one. Things change, players change, rules change, interest rates fluctuate, etc.
Okay. Did anybody get the joke or the allusion in interest rates fluctuate? This is theoretical computer science humor. Does anybody know why I put that in? Okay, just for the fun of it, do a Google search on interest rates fluctuate. All right, continuing though.
So perhaps you should consider some trades to prepare for the changes that might come. You have many choices. Perhaps you suspect that next season's rules will penalize players who do not sing well, so you trade the entire team for the Bach Children's Choir. You have made a change in anticipation of a future that has virtually no chance of materializing, and consequently you will have virtually no chance to repeat as champion. But what if you have an aging veteran who spends most games sitting on the bench? Do you trade for an untested young player with great promise? Perhaps you do. You cannot be sure it's a wise move, but under the right circumstances, it might be a risk worth taking. Each time you gamble with change, you may win, or you may lose. But, if you want to increase your chances of winning, you do not gamble recklessly, you choose your bets very carefully. Successful prene-sets are master gamblers because those that aren't are destined for extinction. Roughly speaking, biological evolution is like a never-ending single elimination tournament, so the fact that HIV exists at all is a powerful demonstration that its current gene team is a champion. Should the owner of the team, the gene set itself, make changes? In this case, we know what the HIV gene set has decided: make lots and lots of changes. In fact, the HIV gene set insists on so many changes that most daughters are mutants of their parent. Can this really be wise? It is, but only because the gene set is very careful about where and how often those changes will occur.
Okay. So I guess a classical view of biology is that when the gene set of some living thing or biological entity, when the gene set reproduces, it's trying to make a perfect copy of itself, and it may occasionally fail. But that's not the case. What it's really doing is it's intentionally making a non-perfect copy of its gene set to adapt to the future. And in HIV, it's quite clear how this works. All right. And it's kind of surprising. So the HIV virus, if you look at one of its daughters produced inside of a host cell, it's not got the same genome as its parent, by and large. Okay. Like, the chances are, let's say, 1 in, let's say, 30, I don't know the actual chances, that the new virus that's just been produced from the old template virus, 1 chance in 30, it's got the same sequence genome as its parent. It usually does not. So is this just that HIV is a lousy replicator? No, it's because HIV wanted it to do that. Okay, so this failure to produce exact copies actually works. It works because
It is, but only because the gene set is very careful about where and how often those changes will occur. Through evolution, HIV has learned that the future is always dismal. Typically, each virus lives in a human with an immune system that is trying to kill it. The immune system is like a trillion-person police force composed of cells that are constantly on the lookout for intruders. From the point of view of many immune cells, an HIV virus looks like a ball wearing a coat made of glycoproteins. The first of these cells to spot an HIV intruder clones itself to produce a multitude of cells, each of which carries a kill on sight poster with a picture of the intruder's coat. The virus is now the hunted. What would you
okay,
do if you were the
Okay, so you follow that? The immune system itself is extremely interesting. And from a purely mathematical point of view, I think it does use what people perceive as randomness, but it's very controlled how it uses it. This has to do with VDJ rearrangement. Okay. But in the end, you end up with this trillion person cell police force that are looking for intruders. And if one of them spots HIV, it will recognize it. One of them might recognize it as an intruder, in which case it'll start to clone itself, make lots of copies of itself. And each one of them will have this picture of the intruder and will try to kill it either directly or indirectly. Okay. So here's the basic question.
What would you do if you were the virus?
What would you do? Well, it says so right there. You would change the code, right? You'd put on a disguise. And that's exactly what the HIV gene set has designed the virus to do. Okay. So here's an example of what goes on. Here's an HIV mother and her daughters at a checkpoint. So the daughters are not identical to the mother. And so in many cases, that means they have a different glycoprotein code than the mother had. And so when the police force encounters the mother, it has this kill on sight poster. It recognizes that this must be killed. But of course, the daughter cells don't look the same. And as a result, they go right through. Questions? Comments? Anything? All right. So how does HIV accomplish this change of glycoprotein codes?
Here is one way HIV does it. The virus stores its gene set in RNA molecules. To increase the copy numbers of the genes, the virus, with the help of its host, makes new copies of itself, including its RNA molecules. The virus uses a wonderful protein called a polymerase to accomplish this.
Have we discussed polymerases before? Someone? Are you guys out there?
I don't think we did.
Okay. Well, I'm glad we did not then. Okay. So polymerases, I'll just read this line.
Polymerase proteins are what life is all about. Without them, DNA and RNA molecules don't reproduce, cells don't reproduce, and you don't reproduce.
Okay. So when we watched that video by Drew Barry, there were some purple widgets that we saw that were making copies. That was an example of a polymerase. There are all sorts of polymerases. In the humans, there are about ballpark 20 different polymerases that are known. Some take DNA and make RNA. Some do various other things, take DNA and make more DNA. Okay. But these are really amazing. So these polymerases are, let's see. Okay. They are really, really small, about 2 million of them side by side would be the diameter of a penny. So they're really, really small. And each of them, they all behave like jugglers on a tightrope. Okay, so what they do, we're going to describe briefly or soon, but they are little teeny machines like we saw in Drew Barry's video that just do amazing things, and they're just nano machines, tiny, tiny things. You know, we scientists can't produce anything remotely as fantastic as these things. Okay, little machines that do amazing things. So here's my image.
Figure 13, the amazing poly, juggler extraordinaire. Think of a nucleic acid molecule as a strand of beads, where each bead can have one of 4 possible colors. A polymerase protein will hop onto a strand and begin to move down the beads. As it passes each bead, it reads the color and uses that as a guide to put a bead of the appropriate color onto a new strand it is stringing. When it is done, the new strand stores the same genes as the original one did, unless the polymerase makes a mistake. One thing humans can be proud of is that their main polymerase almost never makes a mistake. It only puts a bead of the wrong color into the new strand about once in every billion beads.
So the amazing polymerase walks down the template, let's say DNA, where it sees these 4 different colors, A, T, C, and G, really. And while it's walking down it, it is holding in one hand a new strand that it's producing that will have the same information. And if it's reading, for example, a green on the template DNA, it also must reach up into the surrounding solution and grab a green ball to put on the growing new strand. So, of course, there's some occasions when a mistake is made, where the polymerase may be standing on a green base, but accidentally put in a red ball on the new strand. That's a mistake. So exquisite is the human and many other polymerases that it makes a mistake once in about every billion beads. Let's see. Yeah. So every billion beads, it makes a mistake, which is to say, like, close to never. I mean, that's incredible. You go build a little, nanometer size molecule that can do those things. Crazy. It's another example. Nature is amazing. Evolution has created incredible things. All right. So are we good? I'd like to have more discussion and talking. Okay.
Yep.
Yes.
Hi,
Ciao.
Professor. Thanks.
Okay, John.
No worries. Maybe this is broad and I don't want to derail the discussion, but I was wondering what is the role of energy in these computational processes inside the cell. And I think one obvious analogy from today would be with AI systems like large language models and their use of energy, because they are these huge machines which have information stored in them, like prenes stored in physical objects, right? But there is a question as to how energy efficient they are. And so I was
Right.
wondering how energy efficient this
Okay.
natural process is, and if that is a
Yes.
factor in the theory as well.
Yes. And I know the answer, and it's a great question. I'd like to get more great questions. Okay. So when I was doing DNA computing, I calculated all these things, how much energy was being used. And let's say Polly wants to put a green bead on the growing strand. That requires energy, okay, because you're making a change in state of the thing. All right. Now, how much energy does it take? So it turns out that thermodynamics tells us that there's a minimum amount of energy that must be expended to make such a change in state. Okay? And if you look at how much how much energy uh, polymerase uses to make that change, the calculation tells us it's 19 times greater than the minimum required by thermodynamics. All right. Now, 19 times, you might say, well, wouldn't it be better if it just, used the amount required by thermodynamics? The answer is no, because if you use just that minimum amount, then random heat fluctuations in the vicinity of molecules could reverse the process. So the cell has apparently evolved to use energy probably millions or billions of times more efficiently than we do. Okay. Uh, and they could probably have evolved to do even better, but doing that would have left them at the mercy of thermal fluctuations that could undo what they're trying to do. All right. So that's extraordinary. John, what do you think of that use of energy?
That is very interesting because it's almost like metabolism is helping information to endure, right? It's like fighting entropy, sort of.
Yes. Yes, it is. And of course, energy is a very important resource for a cell. I mean, it's got to move around, it's got to make new proteins and stuff like that. All of it requires energy. And so, yes, the cell has to have a way to get energy, and it can't be sloppy with the energy it uses. And it's the opposite of sloppy. It's basically perfect in its energy use. And another example of that kind of thing is uh, photosynthesis. So photosynthesis is a way for the energy to be acquired by plants. Uh, and one could ask, well, if a photon falls on a plant, there's a certain amount of energy in it. Uh, how much energy actually ends up getting changed into chemical energy that can be used inside the cell? And I have not done that calculation, but I'm pretty sure it would be just as amazing as the one I just described. So yeah, the use of energy is amazing. Also, um, along those lines, how efficient is the uh, storage of information in DNA? All right, that is, uh, um, how dense is the information? Okay, so it turns out that if I gave you a 1 inch by 1 inch by 1 inch cube of DNA, which a while ago I used to carry around to make this point, okay, it'll just look like a sugar cube, but a little gooier. And if I gave you that, and of course we know it's storing some prenes are being stored there, and in a base 4. And so we could say, how many bits of information are stored in that little cube? And probably my guess is the answer would be, I mean, I used to know the actual answer. There's a scientific and American article that I wrote on DNA computing that you might look up because I probably worked that out there, or you might look at the original science paper that I wrote, but it's there someplace. And I think my guess is in a 1 square inch, all the information that's currently stored on the internet could go in there. I mean, I haven't looked at that in particular, but that's likely to be the case. So it's another example of evolution's amazing. It does everything perfectly. How did it get so perfect? What was the driving force? What happened to cells that maybe needed 20 times the thermodynamic minimum to do all this when a cell came along and needed only 19 times and could accomplish it? What happened to those cells?
Disappeared, went extinct.
Went extinct. Okay. All right. So thank you for that question. I would thank others for asking other questions like that, because it really gets down to what's going on. Okay. So the human polymerase, the main one, makes only one mistake in every billion beads. That's pretty impressive, right? So let's look at how the HIV polymerase did.
By comparison, the HIV polymerase is pathetic. It puts a bead of the wrong color into the new strand once every couple of thousand beads. Preston, Poish, and Loeb, 1980.
So the human polymerase makes one mistake every billion beads. HIV makes one mistake every about 2,000 beads. And because it's making one mistake every 2,000 beads, and because its genome is 10,000 beads, you can see that most daughter cells are going to have a mistake in them. They're going to be mutants. Okay. So now I get personal.
The HIV virus should be ashamed of its error-prone polymerase. Well, it's not, and it did it on purpose. Why? Because the high rate of mistakes ensures that many daughters have mutated copies of the glycoprotein coat gene and hence different coats than their parent. So, when the immune system's police show up looking for the original intruder's coat, the daughters are already wearing next season's fashion and are allowed to pass unmolested. Eventually, the immune system figures out the deception, begins searching for the new coats, and the process will repeat. When all is said and done, the HIV gene set has used the polymerase gene to ensure a rapid rate of mutation of the glycoprotein gene. The polymerase gene itself has evolved over time, and the current polymerase bears in its very structure the lessons of the past.
Okay, so what we see here is what I had alluded to earlier, that don't think of the genes inside of an HIV genome as being on the same team. They are not. They are enemies of one another. They are struggling to exploit in this case, the virus, to do their bidding. And that means they're struggling to have the virus enter cells, infect hosts, and replicate in such a way that each of them wants their gene to make it through the replication and get to the other side. They want to be happy with replication. And what we see here is the polymerase gene is not itself, well, actually, I can't make this statement quite yet. I believe it's true that we'll see that the polymerase gene does not, when it makes new polymerase molecules, probably does not end up mutating the polymerase gene very much. Okay. But it has caused this rapid mutation in the glycoprotein gene. So the polymerase gene has exploited the glycoprotein gene to mutate, because as a group, they have to mutate, but it has not volunteered probably to mutate itself during this process. Okay. And then this other thing, the polymerase gene itself has evolved over time, and the current polymerase bears in its very structure the lessons of the past. So we can look at the polymerase gene and say, wow, look how sloppy it is. You know, look the way the molecules and the catalytic sites are. You know, it's inviting misincorporation. And yes, it has. But in the polymerase gene itself, it has evolved to have these mutations because it worked. And so the very fact that we can look at it and say, oh, this is a terrible polymerase, is reflecting the fact that that polymerase has learned from the past. Because to be a great polymerase inside of an HIV type molecule is a good way to get killed.
Professor, I just want to make sure that my understanding is correct here. So in this case, like the HIV gene sets exploiting the cell by having this kind of the polymerase prene is exploiting the current kind of like HIV cell.
HIV is a virus, so probably
Oh,
not
sorry,
cell,
virus.
yeah.
To say, well, I'm going to give up this kind of accuracy to increase the mutation rate, and whether that comes at the detriment of any, virus, is kind of not a problem for the pre as long as it can continually kind of evolve, correct?
Well, yes, but it has a really good reason to evolve. If it just sits there and doesn't change its coat very quick, it'll be killed off by the immune system rapidly. All right? By causing all this disruption and this rapid rate of mutation of the glycoprotein, it is evading this trillion man or whatever it is army that is seeking to destroy it. Okay. It has the HIV genome has opted to be very sloppy in order to outrun the immune system, which is the environment it often is in. Did that help at all?
Yeah, I also have one more question. So I know you're talking about the idea that prenes and gene sets don't necessarily have to work together. So my understanding is the polymerase kind of can select to, maybe not select specifically, but maybe target in some sort of way other genes where it's like they can mutate other genes, but not the polymerase itself.
Yes, that would be, I don't have the evidence to say that's actually precisely what's going on in HIV, because I'm not sure whether the polymerase, when it makes new polymerases, when the polymerase gene does that, does it in fact also create so many mutations? I don't know. But my guess is no. So yes,
Okay, that's actually, yeah.
yeah, okay, so and that's the view
Mhm.
of what's going on, right?
Yeah.
And so the polymerase wants to get copied when a new virus is born, but it says, well, I'm not going to let you, the glycoprotein gene, do that because we need to have rapid mutations because of the pressure we're under from the immune system. So we've got to change, but it's you, the glycoprotein gene, that's going to do the changing. We're good the way we are. All right. So, Julian,
Yeah, I just had a question about so I you said that the HIV genome is about like 10,000 like bases long,
bases. Yeah.
and the polymerase here makes a mistake once every 2,000.
Yeah.
So does this mean like if I go in some database right now on the internet and I find the genome for HIV, is this genome entirely different from, let's say, an HIV genome from 1980?
Well, the answer is it will be different, but some of the genes will be presumably the same, and some will be highly different, like glycoproteins. All right. And so, but yes, they won't be the same. You go back and grab a strain, as it's called, uh, in 1980, and a strain that's out there today, going to be a lot of changes.
Yeah. I was just thinking
Right.
there's been billions of copies, probably trillions made of the HIV virus since then.
Oh, yeah. I mean, you know, I guess a ballpark figure would be 100 million people have had this disease, and since the disease does its replicating largely in what are called CD4 positive T cells, about which there's, say, 1 trillion, um, that's a whole lot of people and a whole lot of cells in each of them where HIV is replicating and therefore, because of its lousy polymerase, mutate. And so uh, when that mutation occurs, uh, yes, it may survive the immune system. It may be good at infecting new people. It can have all sorts of advantages, though most mutants don't help. Okay. Uh, so that's sort of the answer. It's not the same. Uh, okay. Any other questions? I really like these questions. Okay.
Well, so,
Yes, Joe.
we've been talking about the lousy polymerase, but it's also worth bringing up the good polymerase because the 1 in 1 billion rate that you described is also highly non-uniform. So it's not just it tries to be the most perfect polymerase and it just makes a mistake every 1 billion
Yes.
beads. Um, there's portions of the genome which are much more likely to mutate than others. And I imagine that if you pin down exactly what they were, they would be things like, the immune genes for the immune system and other things that probably do need to mutate more over time for the same reason.
Yeah, excellent point. So even though the human genome almost never makes a mistake, there's a fairly good likelihood that where it does make mistakes is not just randomly distributed. Okay. That it probably uses those mistakes puts them in very special positions, so-called hotspots. And let me explain how that works. So DNA we can think of as a long chain with A, T, C's, and G's hanging off of it. And that's the so-called primary sequence. But if you take that guy, a piece of DNA with that sequence, and drop it into some salt water, what it does is it changes shape. It tries to, at least in theory, as we've already discussed, achieve the minimum free energy configuration because A's and T's, A's and T's want to be near each other, C's and G's don't, blah, blah, blah. Okay. So these nucleic acids actually have a shape and the three-dimensional shape. And the three-dimensional shape has a lot to do with what happens to them. So when a polymerase jumps onto a strand of DNA, the shape of the DNA itself may make the polymerase more likely to create an error there, or less likely. Presumably, if it's nice and straight in the region currently being transcribed, it probably doesn't make many mistakes. But under the control of the genes, it can have a segment that is all sort of twisted up. And that could lead to mistakes. So that's how the gene set can play with its own nucleic acid sequences to make them hotter to mutation or colder to mutation. I'm not an expert on that, and it's something that immunologists and molecular biologists probably know a great deal about. All right, thanks, Joe. Any more comments? Okay, so
Using polymerase is just one way HIV creates intentional mutations. The configuration, three-dimensional shape, of RNA is largely controlled by the genes. It is easy to imagine that manipulation of this configuration can lead to hotspots where the rate of mutation or recombination is high and cold spots where it is low. HIV apparently creates hotspots in the glycoprotein gene where the rate of recombination is as much as 10 times higher than in surrounding regions, Galeto, et al., 2004. As an aside, it appears that SARS-CoV-2, the virus that causes COVID-19, has a so-called proofreading exoribonuclease associated with its polymerase.
okay. So if you ask, how is it that HIV can be so faithful in its replication of, not HIV, human polymerase can be so faithful in its replication of, say, DNA? The reason is that the polymerase hangs out with other proteins and forms a complex. And among the things it often is seen to hang out with are what are called exoribonucleases. And it's not important what exactly they are, but what they do is they proofread the growing new strand. That is, as the raw polymerase is producing a strand, these things come along and make sure it's doing a good job and proofread it and correct errors. I mean, what can these molecules not do? All right. Now, what does that tell us about the SARS-CoV-2? It's got this proofreading exoribonuclease. And proofreading is a means by which a virus reduces the number of copying mistakes its polymerase makes. HIV has no proofreading. Okay, so SARS, on the other hand, hang on. Yeah. So what does that tell us about SARS, that it has this proofreading? What can we learn from that fact?
It's able to know like the surrounding molecules like DNA and RNA, stuff like that.
Well, let's see. No, if you're going to proofread the polymerase, what you're essentially doing is you're intentionally cutting down the number of mistakes your polymerase might make, right? You're repairing mistakes so that as a complex, the polymerase plus this exoribonuclease nuclease makes less mistakes when copying. Now, HIV made tons of mistakes and had no proofreading. And it did that because it wanted rapid mutation. And why did it want rapid mutation?
Because the environment is the harsh immune system.
Exactly right, John. So that's why it needed it. But now we see that SARS-CoV-2 doesn't take advantage of that. Yes, because it tries to cut down the number of errors it makes. So what does that suggest? Well, the life experience of SARS-CoV-2 is within a host.
It's pretty short because the immune system is trained to take care of it. It's not mutated as much, so it knows how to deal with it.
Exactly right, Julian. That was very good. Yes. All right. So what does that tell us about SARS-CoV-2 approach to staying alive? What would be important if you're not evading the immune system's going to get you. Okay. So what must SARS-CoV-2 do to stay alive if it
It has to find new hosts that don't have immune systems and know how to deal with it. Yet,
Yeah, yes, yet is the important thing. That is, if you can't stay long in the host because the immune system's going to clear you out in a week or two, then it's very important that you be able to spread rapidly, that you have very high infectivity, and that there be competition among variants of the CoV-2 that favors those that have found a way to infect new hosts rapidly. Okay, so here's a statement of that.
By reducing the number of mistakes, SARS-CoV-2 has foregone an important mechanism for avoiding host immune systems. Perhaps the SARS-CoV-2 has other means of avoidance, but if it does not, it will survive only until the host's immune system detects and destroys it, typically within a week or two. Since the virus cannot stay long, to avoid extinction it must infect new hosts quickly. In such viruses, selection may strongly favor mutants with improved infectivity, as has been observed in SARS-CoV-2.
Okay, so that's just an aside. Here's another aside. Okay.
As another aside, since human polymerase almost never makes a mistake, and the damage caused by external insults, such as cosmic rays, is usually repaired. How do humans create variation?
Yeah, so humans do create variation, but they're not, they don't use a bad polymerase to do it. And they repair external insults like cosmic rays. So how do humans create variation?
Isn't it like because um the the mother and the father each contribute 23 chromosomes, isn't there this period where they just kind of randomly swap and it
Yes,
creates a new chain, which makes that's why you're not you and your siblings, you look similar, but you're not exact copies.
perfectly right, Julian Glover. And yes, that we use sexual reproduction to create variation. But now, okay, and so I don't want to review an entire, sort of description of how sexual uh, creation works and the creation of gametes uh, by meiosis and the like. But in the end, uh, you have 23 pairs of chromosomes, and a zygote that's produced, a fertilized egg, um, will now, oh, okay, wait. You're going to get 23 from your mother and 23 from your father chromosomes. And they're matched. There's chromosome 1, you have 2 copies, chromosome 2, 2 copies, blah, blah, blah. But let's take chromosome 1, whatever I'm referring to there. When a sperm is produced by meiosis, you don't actually get just chromosome 1 or its mate in the uh, father, chromosome 1A or 1B, let's call them. What really happens is those two chromosomes line up with one another and they switch part of their uh, sequences. They actually trade, like, I'll keep the top of 1A and the bottom of 1B, or that's called crossover, homologous recombination, whatever. So that's what's used to create variation in humans. Now, the question I want to ask is, do you imagine the cell just does those crossovers at random? Like I take 1A and 1B and I just cross them over at some point and take, the top part of A and the bottom part of B. Do you imagine it could be like that? It couldn't possibly be like that at random, because if you did it at random, uh, the number of possible things you get are astronomical. And sort of like if I took 2 watches and I shook out all the parts of each of them, and they were 2 watches that were not identical. And then I just sort of took pieces from each and put them back together. These 2 watches are not identical. They each ran very well. But if I take pieces, one from each, and I put those together, do you think that clock is going to actually run? Well, if you think about it, the answer is no. So the cell is very careful about where those homologous crossovers occur. And um, so, and they occur at hot spots where crossover is very probable, and what we could call cold spots where it's not. Okay, again, the gene set is controlling its own mutation. All right, so uh, going on, and if you have questions, jump in, please.
Cultural-prene-sets are remarkably precise in controlling how often they gamble and where bets are placed. For example, the human gene set has developed a process called VDJ rearrangement to create a high rate of mutation in the genes that encode antibodies, Wikipedia. Antibodies are shaped like the letter Y. The bottom stem is called the constant region, and the two arms are called the variable regions. The DJ rearrangement virtually guarantees that no two antibodies have the same variable regions, while all antibodies retain the same constant region.
So even on a single molecule, it's probably all really a complex, on an antibody, some parts undergo almost no mutation, and the arms undergo huge amounts of mutation. Okay, Ryan Rubel.
So, if these mistakes that happen sort of at the biological level are intentionally non-uniform, um, to promote some sort of further purpose, because it's beneficial to the population. Do you think that the mistakes that humans make, like at the like psychological level, like just academic mistakes, whatever, do you think those are also non-uniform and that somehow the things that we make mistakes on more often somehow drive the population beneficially?
Wow, I never thought along those lines. So let's think about it a little bit now. Um, Humans make mistakes. Um, that's very interesting. Um, and anybody else who has insights into that. Here's what I'm thinking about, though it may not be very precisely directed at what you say. Let's take when human society produces a new copy of a book. Okay. Now, that new copy may have errors in it, right? Um, it can be just typos. Uh, is that an example of cultural-prene-sets making mistakes? That is, when a society has a printing press and can produce copies that way, uh, does it, most societies, I think, try hard to produce copies that are identical to the original. I would think that's the common thing, but it doesn't always happen, right? So can we go somewhere from that observation and say something about, uh, what Rubel was asking?
I mean, I'm sort of thinking about like what I guess I was thinking about it from the opposite perspective, which is what benefits can we derive from mistakes? So like random example, but I seem to remember that the chocolate chip cookie was a mistake. Someone forgot to fully melt the chocolate.
Oh, yeah.
So I don't know, something like that. Are there beneficial mistakes that we can make?
What I'm having trouble with is seeing that, like in the cookie example, um, that's a mistake and it creates a mutant cookie. And in this case, that mutant cookie was selected, uh, natural selection, I don't know, to go viral. But did the society actually work to create a system where such mistakes would occur. And that I don't know the answer to. Let's ask Kushal Kaladin.
Yes, I think that, I mean, similar to the genetic, on the genetic level, like
Yeah,
you can say maybe society didn't deliberately work, but the genes maybe don't necessarily deliberately work to introduce error. It just so happens that introducing some level of error allows for um, non-exact copies of genes to form and allows for potentially beneficial mutations to occur. So like like penicillin was discovered on accident in a lab because a scientist was doing research on Staphylococcus, and in perhaps it was an accident, and like ideally those kinds of things don't occur. But I think that because of the nature of the way experimentation is carried out, experiment mistakes can occur,
yeah,
and within that lab environment, because the scientist was able to make observations about the experiment and document about the mistake and document what happened, he was able to make like kind of the miraculous discovery. So similarly,
yeah.
like with genes, it could be not necessarily by design, but it's beneficial to have a margin of error in that sense.
Yeah, so I agree that sometimes air can be beneficial. HIV is a great example. Uh, the topic we're discussing right now is a really good topic, and I don't know the answer. All right. But I would guess if you go to Preenland and you think about that question for, say, a couple of years, you will come up with a way of understanding what's going on in those examples we talked about. All right. So, Grayson.
It seems as though in some cases, like society will actively encourage people to make mistakes. Like for example, it seems as though with startups, those are sort of an attempt to like sort of develop the like technological preens that we have. And 90% of the time startups will fail. So in general, founding a startup is usually a mistake, but we've set up our economic system to like incentivize that.
Yeah, that may be a path to understanding this, Grayson. Uh, yeah, so it's a good question, but I do know in my journey through Preenland, when I had questions like that, I could come up with hypotheses about what might be going on and collect examples, which is what we've done so far in this discussion, and see if things worked out. And I found that very often my initial hypotheses looked promising, but fell apart. But with enough time and thought, I thought I got it, right? And that's the things I got were things that I put in these books. Now, uh, yeah, so that's where I think we are. That's an interesting question that's being asked. So, the answer may be buried deep in Preenland. Okay, anything more on that? Okay, so let's see. What time does this class officially end?
350.
Okay, thank you, Len. I'm going to have to leave at 3:40 today. Sorry, but I didn't take a break either, so I'm not that sorry. Okay, now I couldn't resist whoops. I couldn't resist throwing in an additional comment. This is basic, it's not so much about preen theory, but just in general worth saying.
HIV is amazing. A few additional remarks regarding HIV. There are an estimated 35 million humans infected with HIV, so its program for the creation of variation and more generally for survival is succeeding. What can we say about that program? Below is an image of all past presidents of the United States.
Okay. Well, at the time of the writing, I guess, there's Biden is missing from this right now. Okay, but there's, a picture. Okay. And it is stored on my computer as a file of about a quarter of a megabyte. Yes. So here's another picture of it. Yes. But it's a low res picture, and it is stored on a file of just about 2,639 bytes. Okay. And I don't know, I couldn't recognize a single president in that image. All right, so you can't store much in 2,639 bytes, right? I mean, you try to store, something intricate in that many bytes, it degenerates like this picture did. Okay. Now let's ask, what about the HIV genome prene? How many bytes would be needed to store it inside a computer as a sequence of ATCs and Gs, ATCs, A's, C, G's, and T's, thymine and uracil, the interchangeable list. The answer is, since it's only 10,000 long and it's base 4, it's easy to calculate. It comes out to be 2,437 bytes. So HIV, the information in it is less than the information in that image. Yeah, that ain't much. Okay.
Even though it is tiny, the HIV genome prene is the program that creates variation and builds the viruses that infect humans, survive, and reproduce, despite immune systems and drugs. It is a program written by evolution over eons. Our computer industry has nothing that compares. It is worthy of our admiration.
So you go write a 2,500 roughly byte program that does all the things that HIV's program does. And I don't think we have anything that comes close to what the program that HIV is running. I mean, it's a miracle of programming.
The human genome prene requires a file of roughly 750 million bytes, so it is not surprising that we can do and think amazing things.
Right. So if HIV can do this with, 2,500 bytes, what might you expect if you use almost a billion bytes? Well, you should expect a lot. So all you computer scientists out there, including me, I've just insulted, saying that we're not very good at programming compared to nature.
Like all humans, I have many beliefs and these beliefs need not be consistent. See Part 2, Humans and the Gold Star of Truth. I can admire HIV with one set of beliefs and abhor it with another. By adopting a prene-centric view, perhaps I can acquire a useful understanding of the viral enemy.
Yeah, so I actually did some research on HIV back in the day, and it was interesting. Okay, so end detour. Any questions, remarks, whatever? Okay. So the reason for that detour was that the next chapter back in on the origin of societies is called How Cultural-Prene-Sets Program Their Own Mutations. So yes, Catcher.
Basically, this detour, you're saying that our genes basically.
I can't hear you, Catcher. Can you try again, Catcher?
Can you hear me now?
I can.
Great. Basically, I was just wondering. So, just to clarify, this detour is pretty much about how you think that there are kind of mechanisms within our genome and within our cells that can kind of intentionally make mutations to modify proteins in a very specific manner, and that there's a sort of non-randomness to it. Is that somewhat correct?
I think that is very much somewhat correct. Yes, and that I'm saying it's an essential feature of evolution, which I don't recall ever being taught or found in books, that mutation is not random. Mutation is programmed by the gene sets that are being mutated. Okay. And that yes, that are being mutated, right? That's an essential feature of evolution, mutating yourself in a programmed fashion. And okay, so yes, I think you captured a lot of it with your comment, Catcher.
Gotcha. And sorry, just to add on to that, in that case, I think I don't know if you heard of Pierre Alberg, or I don't know how to pronounce his name. He's a German. He did a lot of work in like the 1980s, the 1990s, but he kind of advocates for this internalist perspective of evolution, I guess, or development, developmental evolution. But basically, he kind of his ideas are somewhat similar to yours. I think he has this one paper called "The Logic of Monsters" that I think you'd find really interesting because he kind of postulates that um, there's like internal rules of development that define kind of the possible like phenotypic space of what creatures can form. He has all these examples like teratologies. He uses like, for example, like.
Wait,
Mhm.
check. You're talking about monsters,
Yes,
teratomas.
yeah, that's why he uses like the
Okay.
word monsters. But
Oh,
anyway,
okay.
he's a lot of examples, but I think you'd find it really interesting because it kind of gets at what you're saying in that there's some non-randomness to development and evolution and how forms evolve. Um, it's not necessarily on the genetic level that you're talking about, but he kind of
Yeah.
talks about developmental and a morphogenetic perspective that I think you would agree with or like find interesting.
Okay, so give me the name again, please, of this
Yeah,
thing.
I'll put it in the chat.
Email me. It'll
Okay.
work better. All right, thanks. Okay, there are other hands apparently raised. Okay, so do people have more? Oh, I see, hand raised, yes. Dipsy Desai.
Yes, I had a quick clarifying question. The discussion we had so far, is it also applicable to other types of viruses, or is it more specific to HIV alone?
I think that the general discussion that viruses and other living things, the prene sets have to control their own mutation. That applies to all prene sets in all living things and where other prenes are found. Now, the specific way a given, say, organism will go about programming its own mutation, that can take a zillion different forms. And I think one thing we can say is that any sort of species or culture that is still alive today, hasn't gone extinct, uh, is is likely to have found a good way, at least for now, to program its own mutation. Okay?
Yeah. So I was thinking more from the perspective of computer viruses and worms. I think someone put in the chat that this applies to computer viruses as well.
Yeah, well, um, does it apply to computer viruses? Yeah, I mean, yes, it does. And, uh, the last time I looked at computer viruses, they had become more sophisticated and they had started to behave in ways that resembled HIV. So there are these, antivirus programs, and they're probably more sophisticated than I recall. But roughly speaking, as I recall, they were basically looking for little chunks of code that might be found in a computer virus and making huge libraries of such things. And they would scan your drives and emails and stuff to try to find these sort of lines of code that indicated that you'd been infected. And then they would remove or do something else to those that are found. Is that ballpark right?
I think so.
Okay, so people started to write viruses that would juggle their own code, right? They would, move this subroutine to here and that one to there and blah, 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 and whatever they did. But the virus itself would start to do that. So it was much like the immune system. The antiviral guys were looking for some particular hallmark, like the coat of a virus. And so these newer viruses came out that would try to defeat those attacks, like avoid the immune system, by juggling the parts of the program in such a way that those sequence that identified it as a virus were broken apart and not available anymore. So that to me looks just like what HIV is doing. Okay.
Thank you.
Thank you. Okay. Any more hands? I now figured out how to see when hands are up and stuff. Okay. Are we good? Okay. So let's see where we're at. Okay. All of that journey, detour, was so when we got to this chapter on the origin of societies, how cultural-prene-sets program their own mutation, it would give us a reason to understand why one would ask that question. We now know that programming your own mutations is a critical feature in prene theory, just as replication is. Okay. And so if you're going to study evolution, at least the kind of evolution that we're studying, you have to ask yourself, how is it that cultural-prene-sets go about their programming their own mutation?